Deep underground, in the cold, dark earth, a dormant clove begins to stir. Most plants survive here by acting as passive straws. They pull in moisture and rely on a bustling city of soil microbes to process the nutrients they need to grow. Garlic ignores the standard blueprint. Its roots actively push specialized chemical compounds out into the surrounding soil. This root system exudes diallyl disulfide, a reactive sulfur compound that functions as a natural antibiotic. Since this compound is toxic to many microorganisms, the plant is effectively releasing a disinfectant into the very environment it needs to gather food. Humans have leveraged this chemistry for centuries. During the Second World War, Soviet medics used crushed garlic to treat infected wounds when standard supplies ran low. In 1858, Louis Pasteur observed that even a small amount of garlic juice could stop the growth of bacteria in a petri dish. But unlike a sterilized lab environment, a garden bed requires a living microbiome to function. For a plant that depends on those microbes, these antimicrobial properties present a severe evolutionary challenge. The bulb survives because these chemical secretions are targeted. At the microscopic level, the surface of a garlic root is a zone of constant chemical exchange. As sulfur compounds leak into the dirt, they trigger an oxidative burst. This reaction stunts the growth of competing weeds and repels rot-causing fungi on contact. While red sulfur clouds repel most organisms, threads of green fungus pass through cell walls unharmed. These are arbuscular mycorrhizal fungi, which garlic needs for phosphorus. They avoid lethal sulfur by growing inside the root cells, where concentrations are lower. Garlic maintains a heavily guarded vault, providing a safe harbor only for its essential biological partners. This selection process extends to the bacterial community. The plant actively draws specific microbes toward its root zone. Strains like Proteobacteria flock to the root surface because they have evolved to use that toxic sulfur as a primary energy source. By providing an energy source that only its allies can eat, the garlic bulb suppresses pathogens and assembles a customized microbial workforce. Garlic uses this process to engineer its own rhizosphere, curating the specific microbes that make up its immune system. Zooming out, the reach of this chemical footprint extends well beyond the root itself. This shield often benefits nearby plants. Tomatoes and roses growing within garlic's aromatic radius suffer fewer attacks from aphids and soil-borne pests. However, this protection is selective. Nitrogen-fixing legumes, like beans, are stunted because sulfur interferes with specific bacteria their roots need to function. Planting a row of garlic creates a unified chemical barrier, establishing biological rules for every other organism in the bed. This survival strategy alters the ecology of the entire garden, acting as a guardian for some neighbors and a barrier for others. By midsummer, the leaves begin to yellow. The garlic is harvested, leaving the patch of earth seemingly empty. The soil remains altered. Even after the bulb is gone, the ground is populated by the specific microbial community the garlic recruited. This legacy benefits the next crop in the rotation, which inherits a soil bed that has been effectively cleansed of common pathogens. The humble garlic bulb operates as a calculating architect, shaping the chemistry of its environment long after the harvest is over.